Welcome to the Knowledge Institute. Today's topic is about blood groups. Before we get into blood grouping methods, let's first discuss what is blood. Blood is a connective tissue which is composed of cells and plasma. Cellular components of blood are red blood cells, white blood cells, and platelets, also known as erythrocytes, leukocytes, and thrombocytes. RBC, WBC, and platelets are developed from the bone marrow. Plasma, also called blood plasma, the liquid portion of blood. Plasma serves as a transport medium for delivering nutrients to the cells of various organs of the body and for transporting waste products. The following flowchart gives you a clear understanding of components of blood. In the cellular composition, leukocytes or white blood cells are divided into two categories as granular and agranular. Granular meaning the presence of granules in the cytoplasm of the cell and the nucleus being divided into several lobes. Examples are basophils, eosinophils, and neutrophils. Agranular is the cells that lack granules and nucleus is being unlobed. Examples, monocytes and lymphocytes. Blood plasma contains water, proteins, electrolytes, respiratory gases, nutrients, metabolic wastes, enzymes, and drugs. Moving on to the diagram of cellular components. Red blood cells help in oxygen and carbon dioxide exchange. White blood cells fight against infections. Platelets helps in blood clotting. As you all have a clear understanding about RBC and plasma, we can now move on to our today's topic, blood grouping methods. Human blood is classified into several groups using different blood grouping systems. ABO system, rhesus system, and MNS system. In this video, we'll be talking about ABO system and rhesus system. First, let's talk about ABO blood grouping method. ABO blood grouping system was introduced by Dr. Karl Landsteiner. Landsteiner was an Austrian biologist, a physician, and an immunologist. He discovered the presence of agglutinin in blood. If you are wondering what is agglutinin, agglutinin is a substance in the blood that causes the particles to coagulate, that is, to change the fluid state to a solid state. This method classifies human blood under four main categories as A, B, AB, and O. Now let's take a look at antigen and antibody. For this, you should have a clear understanding of RBC and plasma. That is why we did a small introduction in the beginning of the video. The surface of the red blood cell carries antigen, also known as agglutinogen. There are two types of antigens, antigen A and antigen B. In addition, individuals also carry antibodies in their plasma as anti-A and anti-B. A person with a specific antigen in their red cell does not carry the corresponding antibody in their plasma. If you have antigen A in your red cell, you will not have anti-A in your plasma. The presence of antigen and antibody helps to determine the blood groups of a person. Let's take a look at how blood groups are formed. As we said earlier, a specific antigen will not carry the corresponding antibody in your plasma. For antigen A in the red cell will have antibody B in their plasma. Antigen B in the red cell will have antibody A. If the red cell carries both antigen A and B, the plasma will not have any antibodies. And if the red cell has no antigens, the plasma will have both antibody A and B. So this is how our blood group is formed. The antigen decides our blood group. If we have antigen A, we will have blood group A. If we have antigen B, we will have blood group B. If you have both antigen A and B, your blood group is going to be AB. And if you have no antigens present, your blood group is going to be O. Moving on to the next system, rhesus blood grouping method. Scientists discovered another antigen in the surface membrane of red blood cell. As it was first discovered in rhesus monkeys, it is known as rhesus factor. Rhesus factor. Some individuals have an antigen called a rhesus factor on the plasma membrane of the red blood cell. Individuals having this rhesus factor on their red blood cell are called rhesus positive, and those who lack the rhesus factor are called rhesus negative. Rhesus factor is very important when it comes to blood transfusions. When ABO system and a rhesus system are combined, altogether we get 8 blood groups as A positive, A negative, B positive, B negative, AB positive, AB negative, O positive and O negative. How is blood transfused? In medicine, there are several indications for blood transfusion. When a patient receives blood transfusion, it is vital that they receive blood that is compatible with their own. If it is incompatible, a type of immune response occurs. This is because the donor's cell membrane possesses glycoproteins which act as antigen that react with antibodies, agglutinin, in the recipient's plasma. The result is that donor cells are agglutinated, clumped within the recipient's body. Therefore, it is very important to know the blood group of the donor and the recipient before a blood transfusion. Blood transfusion are categorized into two methods as incompatible blood transfusion and compatible blood transfusion. Let's first take a look at compatible blood transfusion. In a compatible blood transfusion, the donor's erythrocyte antigen and the recipient's plasma antibody are not specific to each other. Therefore, antigen versus antibody reaction do not take place. Therefore, agglutinogen and hemolysis do not occur. Hemolysis meaning breakdown or destruction of the RBC in the recipient's body. So, the transfusion is compatible and successful. In an incompatible blood transfusion, the donor's erythrocyte antigen and the recipient's plasma antibody are specific to each other. Therefore, antigen versus antibody reaction take place. As a result, agglutination or clumping and hemolysis of donor's erythrocyte in the recipient's body take place. So, the transfusion is unsuccessful. Now let's see how blood transfusion occur between different blood groups. Each blood group can receive and donate blood to their own blood groups. Since erythrocyte antigens are absent in blood group O, they can donate blood to all groups. It is known as the universal donor. And as antibodies are absent in blood group AB, they can receive from all blood groups, hence they are universal recipients. let's take a look at blood compatibility along with the rhesus factor. In blood transfusion, rhesus positive individuals can receive blood from both rhesus positive and rhesus negative donors, but rhesus negative individuals can only receive blood from rhesus negative donors. Because when rhesus positive blood is transferred to rhesus negative recipient, he or she develops antibodies against the rhesus factor by immunological responses, and the donated rhesus positive RBC are destroyed by the rhesus antibodies. This makes O negative the universal donor and AB positive the universal recipient. That's all for today, and let's meet again with another interesting topic. Like and subscribe for more. Note any suggestions or questions in the comment section below. Thank you.